Standard engineered systems process information by pushing signals through a rigid physical architecture. The pathways themselves are static. Biological nervous systems operate under a different mechanical principle, continuously modifying their physical structure in response to experience. Biological learning involves a measurable physical alteration of neural tissue. This creates a continuous feedback loop where neural activity drives structural changes through plasticity, and those new structures dictate the path of future neural activity. When an organism masters a motor skill or acquires a new language, the cortex physically reorganizes to support that specific capability. This physical adaptation occurs across all spatial scales, from microscopic synaptic connections to the macroscopic organization of large brain regions. Transient sensory inputs physically sculpt the architecture of the brain. This sculpting begins at the microscopic bedrock of learning, the synaptic cleft. Donald Hebb's 1949 principle is often summarized as fire together, wire together. Biologically, it describes a precise statistical mechanism. Hebbian learning operates as a statistical tendency where repeatedly co-active neurons physically increase their future likelihood of co-activation. The physiological mechanism building this statistical reliability is known as long-term potentiation, or LTP. This animated diagram shows a schematic of a synaptic cleft demonstrating long-term potentiation. We see a persistent increase in synaptic strength following specific patterns of neural activity. The narrowing gap, thickening terminal, and multiplying receptors ensure an identical future signal triggers an amplified postsynaptic response. If synapses only increased in strength via LTP, neural networks would become hypersaturated and unstable. To maintain stability, the nervous system uses a mandatory countermechanism called long-term depression, or LTD. Long-term depression involves the inverse process. Receptors withdraw and the connection physically thins, resulting in a persistent decrease in synaptic strength. Adaptive biological learning requires both construction through LTP to embed regularities and active refinement through LTD to preserve resources and maintain network stability. Plasticity extends beyond isolated synaptic adjustments to the entire physical structure of the neuron. In response to sensory input, neurons physically alter their dendritic branching to seek out new connectivity patterns. Through dendritic arborization, biological cells grow expansive structural tendrils into their surrounding dark environment, searching for new neural pathways. Conversely, dendritic pruning eliminates unused physical branches to optimize network efficiency. Microscopic modifications accumulate into macroscopic reorganizations, such as the thickening of myelin sheaths and the rerouting of white matter tracts. This structural adaptability remains active, supporting skill acquisition throughout the organism's entire lifespan. Biological learning is the multi-scale physical remodeling of the brain's internal wiring diagram. The nervous system must maintain stable representations of complex skills while remaining in a state of continuous physical flux. This stability emerges from the statistical repetition of environmental inputs and behavioral actions. Repeated exposure to a specific visual object consistently co-activates the sequence of neurons responsible for processing its features. This animated diagram illustrates a three-dimensional network, demonstrating how repeated motor actions strengthen the physical links cascading across sensory and motor systems. Recurring patterns become increasingly easier to activate because the physical tissue supporting them has been structurally reinforced. Stabilized pathways physically embed the statistical regularities of the outside environment into biological tissue. The nervous system develops organized structures that reflect an organism's behavioral history while maintaining the flexibility to adapt to novel circumstances. Biological learning is the progressive stabilization of neural organizations through repeated activation and multi-scale plastic modification. Structural learning does not occur within isolated linear chains of individual neurons. The nervous system is organized into interconnected populations whose physical relationships preserve meaningful sensory structures. This topographical map of a neural cortex shows structured zones representing topological maps, such as retinotopic organization for visual space and tonotopic organization for sounds. Plasticity and structural remodeling operate continuously upon these existing topological landscapes. Repeated environmental inputs reshape structural relationships across these vast populations, carving pathways through the overarching grid. Learning produces physical organizations distributed across the network, moving beyond the storage capacity of single cellular locations. These distributed topologies provide the mechanical basis for future, complex neural activity. Learning reshapes macroscopic topological structures, ultimately completing the complex mechanical chain from a microscopic synaptic adjustment all the way to global cortical reorganization. Transient sensory experiences physically embed themselves into biological tissue through continuous structural remodeling. Neural activity operates on landscapes sculpted by experience, which in turn process future information with much greater efficiency. Neural activity influences structure through plasticity. Structure influences future activity through connectivity. The two processes form a continuous feedback loop in which experience gradually sculpts the organization of the nervous system. The result is a biological system capable of constructing increasingly stable neural organizations while remaining flexible enough to adapt to new circumstances. Understanding how these learned structures participate in perception and behavior requires examining how sensory information is transformed as it moves through hierarchical neural systems. This will be the focus of the next chapter.